Welcome back, folks. This is Chapter 3 on cell division, encompassing both mitosis and meiosis. Your goal is to learn in fine detail the underlying processes which drive these two very important cell cycle processes. With respect to the cell cycle, students are expected to know mitosis and meiosis and the difference between them, as well as the similarities, and relate those back into our understanding of genetics. Mitosis is considered a cloning type of cell division because the original mother cell, or the parent cell, is converted to two daughter cells that have exactly the same genetic material. The vast majority of cells in your body are only equipped to perform mitotic cell division. So those cells are known as somatic cells or non-reproductive cells. Humans have pairs of chromosomes, so we are diploid, and the number of chromosomes is 23. Other species will have their own complement of chromosomes. Some species are haploid, they only got a single copy of each type of chromosome. And there are also species on the planet that are triploid, tetraploid, pentaploid, octoploid, etc., etc. Regardless, the duty of mitosis is to take the original genetic material and then make sure that it's divided into the two daughter cells. So naturally, you have to copy it during that process, otherwise you won't have enough to go around. Incidentally, if we have any cells that only contain one copy of each chromosome, then we will use the designation N without the 2 in front of it, and that will be the haploid number. During the production of gametes, eggs and sperm, specialized cells belonging to the germline, which are in themselves 2N, are asked to undergo meiosis, where they halve the genetic information that they then pass on to their daughter cells. Therefore, they go from 2N to N. As part of the meiotic process, the purpose is not to clone the cells, but to produce a reduction in the number of chromosomes, as well as, more importantly, to make sure that the gametes are not genetically identical to one another. Of the 23 pairs of chromosomes that humans possess, one pair are called the sex chromosomes. The others are called autosomes. It is the sex chromosomes that determine the sex of the individual, and these differ amongst the two sexes that we have. Since the types of sex chromosomes are uneven between the two sexes, there are mechanisms which equalize the expression of genes should one sex have more than the other. Incidentally, the discovery of the genes on the X chromosome of humans was the key moment that led to the formation of the chromosome theory of inheritance. Let's focus on mitosis and see how it ensues. One thing that will become clear from the beginning, that it's precisely controlled with lots of quality control steps to prevent an excess of cells being produced and even an insufficient number of cells. The most important thing is that mitosis is a small part of a cell cycle. The cell cycle is a regulated mechanism by which the biochemical changes are coordinated from the moment the cell receives a signal to divide until the point that it does divide. Luckily for us, in eukaryotes, the cell cycles are similar. There are two distinct phases to the cell cycle: a long-lasting interphase, which has a lot of biochemical manipulation taking place inside the cell, and a visible M phase, M for mitosis, that's shorter, and that divides the cells into two after the chromosomes have been segregated. Here's a pictorial depiction of a cell cycle, and the cell cycle will begin here, and then once the cell gets a signal, it'll drop down and enter a biochemical cell cycle. And there are three phases to interphase: the gap 1 phase, where the biochemical content of the cell prepares energetically and in terms of raw materials for S phase. And during S phase, S stands for synthesis, during S phase, the DNA of the cell is doubled. So each chromosome is copied using the DNA replication mechanism. And by the time you get to this point, all the DNA should be perfectly copied on every single chromosome. So the DNA content of the cell doubles. And then the final phase of interphase is the G2 phase, and that stands for gap 2. And once again, the cell is manipulating its biochemistry in preparation for the next phase, which is the M phase, in this case for mitosis. At the beginning of M phase, there are major changes that can be seen under the microscope. Regardless, we'll look at them in a few moments. There are 5 sections to M phase, and each one ends in the word phase. So we have prophase, prometaphase, metaphase, anaphase, and telophase. And then finally right here, somewhere during the point of telophase, the cell actually begins to divide into 2, and that has a name too, that's not listed on this slide, that being the process of cytokinesis. Students are warned to keep a tag on all the different keywords that are being used. Many of them sound similar, but they have completely different meaning. The G0 phase is simply a reference to those cells which are not dividing. A little more detail behind each phase. So gap 1, the G1 phase of interphase, gene expression occurs at a particularly high level because you're making a lot of proteins that are necessary for replicating the DNA, as well as a variety of other factors. During S phase, as we mentioned earlier, the DNA is replicated. Some cells in the body hardly ever divide, such as cardiac cells and neurons, and they are said to be in G0. In the analogy of a racetrack, we have the main racetrack here, and this is the parking garage. So these G0 cells are basically parked in the garage, therefore they're not on the racetrack of cell division. That's a good way to consider G0. But remember, if necessary, the cars can be placed onto the racetrack from the garages. During S phase, the DNA content of the cell is doubled because of DNA replication. Each chromosome that enters the S phase is copied, and the result is 2 sister chromatids, 2 DNA helices held together at some point. These are called sister chromatids. So every single chromosome that enters the S phase at the end of S phase consists of 2 sister chromatids. During the G2 phase, biochemical changes are preparing to engorge the cell in its size, as well as to save up enough energy to permit the separation of the chromosomes, and then finally the division of the mother cell into 2 daughter cells. Cells in G0, the parking garage, are sometimes said to be terminally differentiated. That is, they have reached the end of their specialization, and they're now specialized to be brain cells, kidney cells, whatever. And these cells may not need to divide ever, because there's no need to have extra cells of that type in the body. And therefore, they will eventually perform their functions and die. Recapping what we mentioned earlier, that the M phase is divided into 5 internal subphases. One must also know that the purpose of M phase is to divide the DNA between the 2 daughter nuclei, and that's called karyokinesis. Karyo means DNA, kinesis means separate. And eventually, once the DNA has been separated equally into the 2 daughter nuclei, the cell itself has to be divided, and that's called cytokinesis, the partitioning of the cytoplasm. Here comes the detail behind mitosis. So prior to mitosis, chromosomes cannot be seen. They're too thin and too long to be seen under the light microscope. However, during the first phase of mitosis, which we learned was prophase, the chromosomes are asked to begin to condense by wrapping around themselves and forming thicker and thicker arms. So that's called chromosomal condensation. This process continues until the chromosomes have reached their maximum compactation at metaphase. Each chromosome has a region somewhere along its length that is different to the other regions and the ends. And this is called the centromere, the central part of the chromosome. And these are specialized DNA sequences that don't carry any genetic information, but they have a structural role which becomes clearly evident when mitosis ensues. As these chromosomes condense, they begin to look like X's because each arm is a sister chromatid and they're joined together at their center, the centromere. The centromeres are pretty complicated structures that we have to now learn about. The most important thing is that they bind protein complexes known as kinetocores. We'll be looking at the structure of these kinetocores in a bit more detail shortly. This slide and the subsequent one give us a perspective on the different phases of mitosis with a lot more detail as to what's happening with respect to the nuclear membrane, the centrosomes, and the chromosomes. Students are highly encouraged to reference back into these figures to maintain perspective on what is being taught. We just employed the term centrosomes. Centrosomes are pretty important to learn. They're not centromeres, those are parts of a chromosome. Centrosomes are different organelles present inside animal cells. Plant cells tend not to have these, so they're absent in plant cells. But animal cells, all animal cells have a centrosome, which then duplicates into two before M phase begins. So this happens in G2. One copy then migrates to the other side of the nucleus, as illustrated in the first set of panels. So the centrosomes are in green, lime green. They've already been duplicated, and one of them begins to move to the south side of the cell. And the centrosomes are outside the nucleus at this point. And as they move to the opposite sides of the cell, they generate between them this system of cytoskeleton that we'll talk about shortly. The centrosomes are the origin of the spindle fiber microtubules. So microtubules being one of the three classes of major cytoskeleton proteins. These microtubules have two sides. They have a minus side and a plus side. The minus side is embedded inside the centrosome, and the plus side grows each centrosome in a very coordinated fashion. The result is the formation of a star ray-like structure called an aster around each centrosome. So this is an astra, and it consists of microtubules emanating from the centrosome heart. Some of these microtubules will make contact with the plasma membrane of this cell. Other microtubules will reach across and make a connection with microtubules emanating from the sister centrosome. And a third class of microtubules will make contact with the centromeres of the chromosomes at the kinetochore complex. The kinetochore is a complex of proteins which are designed to allow the microtubules to grow and contract. Thus, the kinetochore machine of microtubules that are present at every centromere of every chromatid are geared towards the movement of those chromatids. The spindle fibers which reach across to the opposite centrosome, the ones that we indicated here, have a name too. Those are called polar microtubules. And their job is to grow and push against each other, therefore elongating the cell as well as stabilizing the internal structure responsible for separating the chromosomes. And we already spoke about the astral microtubules, and they grow away from the centrosome towards the plasma membrane. Thus, once the chromosomes have condensed and the nuclear membrane has melted away into the cytosol, and the spindle complex has formed, we have proceeded through prophase and prometaphase. The next phase is called metaphase. The sole purpose of metaphase is to line the chromosomes up in the center of the cell so that they can be appropriately separated. In order to prevent tangling and breakage of chromosomes, which are really quite fragile, the metaphase chromosomes are condensed to their maximum, and thus 10,000 times more folded than they were during interphase. By pulling on the chromosomes from opposite sides, the kinetochore microtubules are able to position each chromosome at the metaphase plate. The metaphase plate is nothing more than a hypothetical region between the two centrosomes at the equator of the cell. One may ask, if sister chromatids are being pulled in opposite directions, why don't they separate? The answer is that they are attached to each other using a molecular adhesive, which in this case is a protein. And the name of that protein is cohesin. So sister chromatids are held together by cohesin, which resists the tension created by the pull of the kinetochore microtubules. Cohesin is a complex protein of 4 subunits that coats the sister chromatids along their entire length, with the greatest concentration being found at the centromeres, probably because that's where most of the tension is. In figure 3.4, the use of these orange dots is meant to symbolize the presence of the protein cohesin. The next phase after metaphase would be anaphase. And anaphase is characterized by the separation of the sister chromatids. And that takes place because the cohesin is destroyed by an enzyme called separase. So separase separates the sister chromatids, and that happens in the early phase of anaphase, known as Anaphase I. The genetic term applied to the separation of the sister chromatids is disjunction. So here we have a pair of sister chromatids, i.e. a single chromosome, undergoing disjunction. The force responsible for separating the 2 sister chromatids is applied by the pulling on the kinetochore by the microtubules. So the way this takes place is that the microtubules that are attached to the kinetochore begin to undergo depolymerization. And that depolymerization results in their ends shortening, therefore being pulled towards each centrosome. Incidentally, within each organelle called a centrosome, there are 2 centrioles, just so as to confuse you with all these centro somethings. Subsequently, in the second half of anaphase, known as Anaphase B, the polar microtubules begin to lengthen, therefore pushing each aster away from its partner. That lengthens the cell, allowing more space for cytokinesis to get things right. The final phase of mitosis is characterized by telophase, where the nuclear membranes begin to rematerialize, reassemble, around the newly segregated chromosomes at each end of the cell. And the beginnings of cytokinesis, which in animal cells is demarcated by a cleavage furrow, like a belt tightening, and in plant cells by the formation of a brand new cell wall between the 2 daughter cells. In animal cells, that cleavage furrow is actually a second type of cytoskeleton protein called actin that makes a contractile ring, and the actin will constrict, forming the cleavage furrow. In both animal cells and plant cells, cytokinesis will actually divide the cell in 2. The number of organelles is not really controlled directly. They happen to just lie randomly, and hopefully by chance, because their numbers are so great, some will be captured by each daughter cell. If everything goes as planned, the result is that each daughter cell should have 1 copy of each of the original DNA double helix. There should be no loss or gain of genetic information compared to the parent cell, the mother cell. Each daughter cell should be 2N, and therefore that can be maintained consistently through future generations. The terms chromosome and chromatid need to be applied appropriately, otherwise there can be confusion. Whenever a single entity exists, it's called a chromosome. So in interphase, each double helix, each chromosome, which looks like an I in G1, is a chromosome. But during S phase, each of these chromosomes is replicated, but not detached from its copy, and it begins to look like an X. But each is still a single entity, is called a chromosome, even though it consists of 2 sister chromatids. So keeping that in mind, we can then relate to this mathematics. So the number of chromosomes consistently stays at 46, the number of individual entities of DNA. But the number of chromatids doubles in S phase, even though they're still held together to form 46 chromosomes by the centromeres. Once mitosis is complete, the total number of chromatids is reduced down to 46 because each one is now not an X but an I-shaped DNA double helix. If somebody is measuring the amount of DNA in a typical nucleus, then we can see that there's a doubling of the amount of DNA before it's being reduced back to the original amount. And that's the purpose of S phase, to copy the DNA so that it can be divided amongst the two daughter cells equally. In summary, mitosis is a coordinated sequence of events which results in the formation of two identical daughter cells, which are then identical to the original parent cell. First, the chromosomes are duplicated, and then the cell is cut in two. We mentioned earlier that this is a very, very coordinated sequence of biochemical events, which results in anatomical changes for the chromosomes as well as the cell itself. Biochemically, there are many quality control steps built in between each phase. These are called checkpoints. Checkpoints are important because this is an opportunity for the cell to chemically check if things are going correctly. Since a single cell doesn't have a brain, how does it know what the answers to these questions are? Well, amazingly, we use proteins and biochemistry to answer these questions. So protein interactions are responsible for ensuring the answer to the quality control questions. If things go right, the protein interactions lead to one pathway. If things go wrong, then these protein interactions are different and they lead to alternative pathways. In addition, very importantly, if the checkpoint proteins themselves are defective, then there's a possibility that when it comes to the checkpoint, it won't be performed properly, and that will lead to abnormal results. Indeed, abnormalities are common in cells, and some cells do fail these checkpoints, and the outcome can be one of two things. It could be cell death, or at the other end of the spectrum, we can have something for a multicelled organism, such as animals, cancer. Cancer is characterized as cells which no longer obey checkpoint rules and grow out of control. Many checkpoints have been found, and more are being discovered all the time. The major ones seem to lie at the positions indicated on this figure. A master checkpoint is called the G1 checkpoint, which lies some distance into the G1 phase. This checkpoint has a two-point purpose. It can allow a cell to proceed, or it can return a cell back into G0. If a cell is passed through the G1 checkpoint, then either the cell will pass all the other checkpoints and emerge as two daughter cells, or if it fails these checkpoints, it will be given one last opportunity to fix the error before it's killed by apoptosis. These subsequent checkpoints are noted for certain features. The S-phase checkpoint is designed to repair any base pair mismatch errors that may have taken place during DNA replication. The G2 checkpoint makes sure that all the DNA, all the chromosomes have been copied completely, and that the cell has adequate shape and size, space, to continue into M phase. The metaphase checkpoint is designed to make sure that all the chromosomes are lined up at the mitotic spindle. Checkpoints are very, very important. Let's move now on to meiosis, a type of cell division only performed by a tiny fraction of your cells in your body. Meiosis is reserved for the germline cells. It results in 4 daughter cells, not 2, and it has 2 types of cell division, not 1, and the DNA is shuffled to make sure that it is as different as possible compared to the parental cells. Just to remind you that in the world of biology, there's 2 types of reproduction. There's asexual reproduction for organisms that do not need a mating partner, and there's sexual reproduction where gametes are formed by each partner and 2 parents are needed. The gametes will subsequently unite if mating ensues, to form a offspring, and that process of one gamete meeting the other is called fertilization. The entire purpose of meiosis is to produce haploid gametes. Haploid gametes. They should contain half the genetic material compared to the parent cells. Although different terms may be used for these gametes when they come from male versus female, they have exactly the same format. So for animals, for male, we call them sperm. For plants, we call them pollen. As you'll become aware during the preceding few slides, there are similarities and differences between mitosis and meiosis. Paying attention to these will help you recall a lot of this information. Some of the major differences are as follows. In mitosis, we begin with a single parent cell and we end up with two daughter cells. In meiosis, we start with a single parent cell and it undergoes two divisions, as well as producing not two but four daughter cells. If one looks a bit more closely, you'll notice that the number of chromosomes is reduced in the daughter cells compared to the parent cell. The most fundamental thing to remember is that DNA is only replicated before the two cell divisions take place. And there's only one phase at which DNA is replicated. So we take a journey back to the diagram showing the cell cycle. If we were to reinterpret this in the case of meiosis, we would start over here. We will travel down through the three phases of interphase, copying our DNA here, and then we will take a journey through this segment once, then return back to the beginning of this segment and take a second journey. And then we will end up first with two daughter cells doing our first journey, and then each of those two daughter cells will return to this point and take a second journey, ended up with four daughter cells. But you only go through this phase once. That is key. Table 3.1 does a great job of contrasting mitosis with meiosis. From the genetics perspective, there's one pink elephant in the room, and that elephant is the difference between mitosis and meiosis. In mitosis, the homologous chromosomes do not interact with each other whatsoever. In meiosis, the homologous chromosomes are meant to come together in meiosis I and pair up. Overall, meiosis has three major hallmarks. The first is what we just discussed, that homologous chromosomes pair up. The second thing is, once these homologous chromosomes have paired, they are now in a position to physically exchange DNA. That's called crossing over. And then the third hallmark of meiosis is that the homologous chromosomes are segregated from each other at the first phase of cell division. Because of the more complicated nature of prophase, we have expanded prophase I into sub-stages. So we call those sub-stages prophase I leptotene, prophase I zygotene, prophase I pachytene, prophase I diplotene, and prophase I diakinesis. The first phase, leptotene, is characterized by two events. First is that the chromosomes begin to condense. And the second thing is that the meiotic spindle begins to form microtubules that extend from the centrosomes. That's it. Then zygotene is characterized by the nuclear envelope melting away, disintegrating, and the movement of the homologous chromosomes as they begin to shorten to form synapses, pairing. These synapses are pretty complex in themselves. So as the homologous chromosomes align with each other based on DNA sequence, they then are recruiting proteins to form a bridge between them. That's called the synaptonemal complex. It's a complicated mix of three protein layers that is designed to identify non-sister chromatids and bind them together. So the important thing is, it's never been known to bind sister chromatids together. It always picks non-sister chromatids and binds them together. This time-lapse illustration indicates what happens in different phases. If you look up and down as columns, they refer to the phase below. So this is interphase, and then this is leptotene, zygotene, then pachytene, and diplotene, and diakinesis. We can see the synaptonemal complex begins to form in late zygotene and continues to function during pachytene and the early part of diplotene, and by the end of diplotene it begins to dissociate. The proteins are present here, and those proteins hold the DNA together between two non-sister chromatids. And the presence of this green dot indicates a recombination nodule, a recombination machine that's present on the surface between the two DNA molecules. And this is responsible for precisely cutting and rejoining the DNA between the two non-sister chromatids. In pachytene, the chromosome condensation continues, and the homologs are now so close together that the two sister chromatids of one homolog and the two sister chromatids of the other homolog, as well as their synaptonemal complexes in place, form something known as a tetrad. And these tetrads are visible under the microscope, and they appear to have 4 chromatids. The recombination nodules, the green structures that we spoke about on this figure here, are responsible for crossing over. By diplotene, crossing over has completed, so that the synaptonemal complex is no longer needed and it begins to dissolve. The homologs are pulled apart slightly, revealing the existence of points of crossing over known as chiasmata. And these are typically present in numbers that are pretty low, 3 to 5 per chromosome. Once more, cohesion protein is found between sister chromatids, and that resists the forces of the kinetochore microtubules in the same way that we saw with mitosis. The entire tetrad is moved towards the metaphase plate during diakinesis. Figure 3.12 illustrates these events in Meiosis I. Compare this to what we saw when we were looking at mitosis, and you'll see a stark difference. Here we just have one homolog. In the example that we just looked at in meiosis, we appear to have 2 homologs. As the cell cycle moves into metaphase I, the chiasmata that were holding the homologs together have been resolved, and the crossing over events are now completed. Homologous chromosomes align on opposite sides of the metaphase plate, but independently of neighboring chromosomes, which line according to their own orientation. The kinetochore microtubules, which are attached to both sister chromatids of one homolog, point in one direction, and the microtubules at the kinetochore of the other homologous chromosome and its sister chromatids attached to the opposite pole, so that chromosomes are pulled away from their homologs. Anaphase I shows the separation of the homologous chromosomes from each other. Remember that the sister chromatids of each homologous chromosome are still attached to each other by cohesion, so that has not melted yet, but the homologous pairs are separated from each other. The cell then enters telophase I, where the nuclear membranes begin to reform around each of the 2 halves of the cell, and the cell begins to divide into 2 by cytokinesis. If one studies the ploidy of the cell with respect to the chromosome count, we started with 2n before meiosis I. At the end of meiosis I, each daughter cell will have only n number of chromosomes. When it comes to pairing up chromosomes, the autosomes have no problem finding their homologs because they have a lot of resemblance in their DNA sequences. But how about the sex chromosomes? We know that the X chromosome is far larger than the Y chromosome, and they carry different genes. Recent research has shown that the ends of the X and Y chromosome carry enough genetic information in common to be able to form a synapse during prophase I in males. Remember, it doesn't need to happen in females because females will then have 2 X chromosomes. And the synapses of the X and Y is possible because of these pseudoautosomal regions at the ends of the 2 sex chromosomes. That's illustrated quite nicely here. So this is par 1 for the X chromosome and par 2, and this is par 1 for the Y chromosome and par 2. Please keep a note on this gene called the SRY. Once meiosis I is complete, each of the 2 daughter cells has a small pause in some cases, but in others none, and then it moves on. Each of them moves on to perform meiosis II. Recall, even though we have reduced the ploidy of the daughter cells to half that of the parent cell or the mother cell, each chromosome still consists of sister chromatids, and that's not normal. So in meiosis II, the sister chromatids will be separated from each other so that each haploid daughter cell that will emerge will have just 1 chromosome of each type. And because of the process of crossing over and other things that we haven't really considered, the 4 daughter cells that will be generated overall will be genetically distinct and haploid. In many ways, meiosis II has resemblance to mitosis. Please study these panels to get a full indication of what happens during each phase. Relating the cell cycle to what Mendel observed when he was conducting his experiments and Mendelian ratios that emerge from simple genetic crosses, we find that one supports the other absolutely. If a heterozygous parent carrying, for instance, big A and small a, enters into meiosis, his or her gametes will contain either big A or small a in a ratio of 1 to 1. And this now happens, according to our understanding of meiosis, at Anaphase I. So Mendel's law of segregation applies at Anaphase I. Figure 3.15 uses big G and small g to indicate the same point. So this cell came from, say, the mother. She was heterozygous. Then she'll end up performing meiosis on the germline cells, and these are her eggs, and they will contain either big G or small g. And because she produces a couple of million eggs, the ratio of the 2 will be approximately half this and half that. To implement and understand Mendel's second law, that of independent assortment, we have to expand our understanding to multiple chromosomes on different homologs. For instance, we could have chromosome number 1 and chromosome number 2 in humans. So the way these chromosomes line up during metaphase I is very important. In metaphase I, they could line up in either orientation with respect to each other. So you have the father's chromosome 1, it could line up on the same side as the father's chromosome 2. Or it could line up on the opposite side of father's chromosome 2, such that the mother's and father's chromosome 1 and 2 are on the same side. Figure 3.16 helps us understand this. Assume this is the father, and the father has 2 sets of chromosomes. So we have one set of chromosomes carrying the G gene, and then the smaller set of chromosomes are carrying the R gene. And in each case, he's heterozygous, a double heterozygote. As meiosis takes place in his testes, the germline cells will be then possible to segregate the chromosomes in 2 fashions. The first is where the chromosomes line up such that the big G and the big R are on the same side of the metaphase plate. That's one possibility, and that will happen in about half of his sperm-producing germline stem cells. The other half is the alternative possibility, in that, once again, the big G is lined up on the left-hand side, but this time the big R is lined up on the right-hand side, compared to this example. And this will happen in the other half, randomly, of his cells. The overall result is that when he produces sperm, millions and millions of sperm each day, they will have a roughly 50-50 relationship between the two orientations in which the chromosomes have lined up. Because each chromosome is an independent entity, it has no respect for the lining up of the other pair. The other pair can do what they like, either this direction or that direction. Therefore, it is possible for him, with 2 pairs of genes, to have 4 gametes, 4 combinations of gametes. So if you study this figure, you will find that this is 1, this is the 2nd, this is the 3rd, and this is the 4th class of gamete. If one pathway was favored more than the other, then all the gametes may be just of 2 types, and that would then change the ratios that Mendel was able to observe. But that's not the case. This is a random process designed to assist in genetic variation. We now move on to another section of the chapter that tries to reinforce some principles of genetics that are assumed under various hypotheses. The first of these is the chromosome theory of heredity. That proposes that genes are carried on chromosomes. So scientists such as Sutton and Boveri, they proposed that hypothesis, and they were able to confirm it by following chromosome behavior in meiosis and then translating that into hereditary transmission of traits, genes, and their alleles. Experiments by Morgan confirmed the chromosome theory of heredity. He was working on Drosophila melanogaster, which are the fruit flies that you see flying around the bananas. He did this by studying the eye color of these flies. Before we move on to look at the experiment, it's important to understand the term wild type. The wild type doesn't refer to any particular phenotype, but what it does refer to is the most common phenotype in that particular population. So, for instance, if we looked at human populations, the wild type eye color in Africa would be black, whereas the wild type eye color in Sweden would be blue. With Morgan's experiments, his laboratory were breeding these flies continuously for a number of years, and during those years they discovered numerous phenotypic variants or mutations, and they analyzed the crosses that produced these variants, and then they used the variants to perform further genetic crosses to deduce genetically what was happening. Before we consider his experiments, let's go straight to his conclusion. He concluded that the results were consistent with the hypothesis that genes are carried on chromosomes. During this period of Morgan's experiments, another scientist published her data. Natalie Stevens was working on beetles, and by carefully observing the cells of these beetles, she discovered that the female beetles contained 20 large chromosomes. However, the males only contained 19 large chromosomes, plus a tiny small one. So her studies indicated that there was a difference in the size of the chromosomes pertaining to sex in these beetles. In 1910, Morgan published a paper which validated Stevens' conclusions. And from that, we now know that there are 2 references to sex chromosomes. Something can be sex-linked. It simply means that the genes are resident on one or the other of the sex chromosomes, and that the pattern of transmissions is sex-orientated. And the second term is more refined. It refers to X-linked inheritance and Y-linked inheritance. In X-linked inheritance, the genes are on the X chromosome and not the Y chromosome, and in Y-linked inheritance, the reciprocal case applies. The very first mutant that the Morgan lab identified was a white-eyed male. The fly had a white eye compared to the wild type red eye. So this is a red-eyed fly, male, and this is the mutant fly. It has a white eye, phenotypic. The lab mated the white-eyed male with a normal red-eyed female, and they looked at the F1 progeny. And all the F1, all of them had red eyes. From these findings, they concluded that the white-eye mutant allele was recessive. They then bred the F1 flies together to produce the F2 flies. And as expected for a recessive allele, the white-eyed flies were regenerated in the F2. But unlike Mendel, the numbers were off. They failed to obtain the 3:1 ratio they were looking for. These are the actual numbers that they obtained, with the following very important observation. Only the males had white eyes. Eventually, they were able to obtain white-eyed females, and when they crossed those white-eyed females with wild-type red-eyed males, they produced red-eyed females and white-eyed males in the F1, but no white-eyed females. But when they self-fertilized the F1, they were able to produce in the F2 both red and white-eyed flies with equal frequencies among both sexes. That reciprocal cross, that alternative cross, breeding the mutant in the opposite direction, produced the data as indicated here. These unusual shifting patterns between the sexes indicate sex-linked traits and gave a clue as to the biochemical nature by which sex is determined in these flies. This is a clear departure from Mendel's results, who had not had to contend with sex-related conditions in his peas. Let's add in the genetic notations for males and females. So females have a upside down cross circle and males have an erect arrow. So the females would have two X chromosomes, the male would have an X and a Y. The Y is empty with respect to this particular condition, so we leave it empty. So if the male has the mutation, then we can use the small w to indicate white. And using X chromosomes with W plus indicates the wild type allele, the red allele. So this female is bred to this male and they produce the F1. And the F1 are all red-eyed because the male has inherited from his mother the wild type X chromosome. The female, however, has become heterozygous and is said to be a carrier. When the F1 are crossed with each other, we see the gametes formed for the males on top and the female eggs on the side. And it's simply a matter of predicting what we would see in the F2. And in the F2, we divide it into two columns. The first column is the column for females, because it contains two X chromosomes. And the second column is the column for males, and that contains only one X chromosome with a Y. So all the females will be red-eyed, and the males will be red-eyed and white-eyed in a 1:1 ratio. Even though the females are all red-eyed, they are also divided into carriers and homozygous dominant, and that genotypic ratio would be 1:1 also. Morgan's work supported the chromosome theory of heredity. Calvin Bridges was also studying fly eye color phenotypes and looking for abnormal chromosome numbers. Every so often he would find something unexpected, maybe at a ratio of 1 in 2,000. He found females with white eyes or males with red eyes when he wasn't expecting them. By studying these individuals further, he saw that the exceptional females had two X chromosomes and a Y chromosome. So they had 3 chromosomes. And these exceptional males had just one X chromosome and no other chromosome. So they were just X, 0. Bridges concluded at the genetic level what the reason for these errors was. He came to the conclusion that the chromosomes were being separated in an unusual pattern during the formation of gametes. And when that happens, when chromosomes are separated in an unusual fashion with one chromosome going in the wrong direction, we use the term non-disjunction. Remember, we use the term disjunction before. Now we use the word non-disjunction when the chromosomes don't obey the direction in which they're supposed to move. The unusual genetic crosses are indicated here. So here's a normal female and a normal male. The male is wild type, the female is a double white mutant. He, in this example, produces normal gametes as we expect in his sperm, but she has some weird, unusual non-disjunction going on. So these two chromosomes in meiosis are supposed to segregate one in each direction, but both these chromosomes accidentally get pulled to the same pole in meiosis I, and that results in no chromosomes present in these eggs and extra chromosomes present in those eggs. And this will then influence how the genetic makeup of the offspring occurs. So in this case, we have 3 chromosomes present, 3 X chromosomes present in the zygote, and that is lethal. And when we only have the Y chromosome coming down from the father and no chromosomes coming from these eggs, that's also lethal. So these 2 boxes will not produce any offspring. But the other 2 boxes, the diagonal boxes, will produce offspring. So males can have 2 X's and a Y, and females can be XO. Those results were key in allowing Bridges to conclude that sex determination in these flies involves genetic and biological processes, and that the male and female characteristics of a species are the result of the interaction of both. However, it's not that simple. You can be born as a male and develop into a female, because what you inherit in terms of your genes is your potential. Then what is expressed from those genes and other environmental interactions may not manifest in you developing into your genetic sex. So we have something known as chromosomal sex, and then we also have something known as phenotypic sex, and there's a couple of other categories that we'll talk about in a future chapter. So chromosomal sex is simply the chromosome complement that you inherited between the union of a male and female gamete. So that happens at the moment of fertilization, conception. Phenotypic sex comes a bit later. That's the result of interaction between the chemistry and the environment, the biochemistry and the environment. So these could be the result of you having a gene for making you male, but if that gene is broken or not turned on at the right time, you may not develop into a male. Thus, phenotypic sex has to do with gene expression at a later point than in chromosomal sex. Sex determination in these fruit flies, Drosophila, is different to sex determination in humans. Females who have 2 X chromosomes will become female. Males who have 1 X chromosome will become male. That is the rule. So applying this rule to the examples that we see in previous crosses, we would expect anything that with a single X chromosome would develop into a male, regardless of what else they have. Anything with 2 X chromosomes would develop into a female, regardless of how many Y chromosomes they have. So an X, an X, and an X, male. 2 X's, 2 X's, female. Since zygotes with a YO have neither X chromosome, they will never be seen, because you must have an X chromosome to survive. And females that have 3 X chromosomes are very rare, although they are found. There is something known as the X to autosomal ratio. For each autosomal chromosome, what number of X's do you have? So the XA ratio is able to determine the gender based on the number of X chromosomes to the number of sets of autosomes. So in the example of the males, the XA ratio is 0.5. And for females, the ratio is 1. So males have half the dose of female X chromosomes in their cells. With the placental mammals, mammals with a placenta, the situation is slightly different. We also have X and Y chromosomes, but our sex determination comes in a slightly different fashion. We have on the X chromosome about 1500 genes. On the Y chromosome, we have just under 50 genes. One of those genes on the Y chromosome is very important. It's called SRY, for sex determining region of Y. This one gene is responsible for determining the sex of the individual. If this gene is present, then that individual will be male. If the gene is absent, that individual will be female. If one looks across this row, you will see that there is at least one Y, and that will make that individual male. In the row below, there is no Y, and that individual will be female. It turns out that the SRY locus produces a transcription factor which is needed to activate a bunch of other genes that are responsible for making males. The relationship between genetics and developmental biology has now been concluded successfully. It turns out that in these placental mammals, the embryo is a cluster of tissue with undifferentiated gonads, which can develop in the future into either ovaries or testes. The expression of the SRY gene initiates a program of development that turns this undifferentiated tissue into testes. In the absence of the expression of this SRY gene, the default pathway is to develop into a female, and that's indicated here. So this is the default pathway, and this only takes place if that gene is activated. Now you can see what could happen. Somebody could be genetically XY, but they could have a mutation in the SRY gene and not activate the transcription factor, and therefore they will develop this way. They will be anatomically female but genetically male, and that's causing a lot of controversy right now with the Olympics committee, because there are some runners that belong to this latter category. An alternative system is used by some animals. That system is known as the ZW system. So many birds use it, not all. Some reptiles, some fish, all butterflies, and all moths. The system relies on the fact that females have two different sex chromosomes and the males have the two equivalent sex chromosomes, thus the opposite to us. See if you can understand the designation of the ZW system by following this genetic cross. Are you able to correctly predict the genotype of these two individuals looking at just their phenotype? Please have a go. Now do the same for this reciprocal cross where the parents are genetically reversed. Do you get the right result? Because of the way the sex chromosomes are inherited, in humans, sex-linked trait transmission follows distinct patterns. So the first thing to realize is that if the trait is X-linked recessive, then females who are homozygous for the recessive allele will express it, and only males that are hemizygous will express it. Keep this in mind when studying pedigrees. If the trait is X-linked dominant, females that are homozygous dominant or heterozygous will express the trait, whereas males who are hemizygous will always express the trait. Hemizygous males display any allele on their single X chromosome, regardless of whether it's recessive or dominant, because it has no opposition. Any trait on the Y chromosome will always be passed from father to son and son to grandson, never into the female unless there's some type of mistake. Inheritance of that type is called patrilineal. A typical telltale sign of X-linked inheritance is whenever you see in a pedigree or in a genetic cross the expression of that trait more often in males than in females. Humans have a lot of genes that are X-linked because the X chromosome is quite large and it carries, as we said earlier, about 1500 genes. The most common two are listed here: color blindness, red-green, and hemophilia A. But as you can see so far, we've discovered a whole bunch more of dominant and recessive conditions that are inherited on the X chromosome. The movement of chromosomes carrying the recessive allele can give rise to a phenomenon of generation skipping. In that example, a grandfather will pass on his X chromosome to his daughter, and the daughter may pass on that to the grandson. So the daughter doesn't have it, but the two males in the generations above and below do. And as we've already seen with the fruit flies, a recessive male, when mated to a homozygous dominant female, produces all offspring with the dominant phenotype. But the females are carriers. Carrier females, when mated with recessive males, will produce about half their offspring carrying the dominant condition and half carrying the recessive condition for both sexes. This is easy to explain because the females are heterozygous. One of their chromosomes carries the wild type, the other one the mutant, and depending on which male gets which, they will have a 1-to-1 ratio of disease versus normal. Likewise, homozygous recessive females, females with the condition, when bred with dominant males, will produce all dominant carrier female offspring and all recessive male offspring. Here is a typical pedigree showing an X-linked recessive inheritance pattern, in this case for red-green color blindness. Red-green color blindness afflicts males much more commonly than it does females, about 400 times. A famous example of an X-linked recessive trait that nearly all genetics textbooks present is that of Queen Victoria of England, and a new mutation that took place in her called a de novo mutation. She didn't have the disease, but somewhere during the production of her eggs, the mutation must have appeared. The mutation is known as hemophilia A, and it's a mutation in a gene that produces a protein known as factor VIII. Factor VIII is very important in blood clotting. Individuals that have a defective gene will lead to bleeding and death. The European royal family pedigree, in addition to showing inbreeding, it shows the transmittance of this mutation that was first developed inside Queen Victoria. See if you can apply some of the rules you just learned over the last few slides to this one to predict what will happen. Some alleles function in a dominant fashion. X-linked dominant traits are known, and when we study them, we find some distinctive characteristics of that type of transmittance too. One rule is that heterozygous females, big A, small a, when mated to a recessive male, small a, transmit the dominant allele to half their progeny of each sex equally. However, if dominant males, big A, are mated to homozygous recessive females, small a, the females pass the dominant trait to all their daughters and none of their sons. A famous example of an X-linked dominant allele is something known as congenital hypertrichosis, also colloquially known as werewolf syndrome. It causes the afflicted individual to have extra hair follicles all over their body that produce hair continuously. And the affected males or females have more body hair than normal, as you can see here. This actual pedigree shows how the female passed on this condition to two of her children, and then how they transmitted it to some of their offspring. But you'll notice something strange here. This male only passed it to his daughters, and he had 5, but not any of his 4 sons. Let's turn our attention to Y-linked inheritance. As we mentioned earlier, Y-linked traits are only transmitted exclusively from male to male. We also mentioned earlier that mammals have fewer than 50 genes on the Y chromosome, and these genes are most likely involved in some type of sex determination or development. And that's true. So far, none of the genes on the human Y chromosome have a copy on the X chromosome. The final topic, and maybe one of the most important in this chapter, is that of gene dosage compensation. Let's start by telling you that in males, they have one X chromosome, and that chromosome has about 1,500 genes. The ratio between that X chromosome and all the other autosomes is one autosome set to one X chromosome. And that's always going to be the case as far as males are concerned, because they normally have one X chromosome. But females have two X chromosomes. So that ratio becomes abnormal. And one solution that nature has is to, very early in development, shut down randomly one of the X chromosomes in that cell. Why? To prevent the ratio from becoming upset, and therefore the biochemical activity within that cell will be out of sync. This compensatory mechanism is known as dosage compensation. So far, 4 different mechanisms have been discovered, which are presented in Table 3.3. Please make yourselves familiar with this. In fruit fly experiments, we now know what happens. Because males only have one X chromosome, that X chromosome is expressed at twice the rate in males as it would be in females. So there's your compensatory mechanism. Increase the rate of expression. In roundworms, the gene expression of each X chromosome in the hermaphrodite female is decreased to half the rate of that in the male. So this is the opposite situation compared to flies. In marsupial mammals, the paternally derived X chromosome is inactivated in all female somatic cells. So the father's X chromosome, when it enters into the zygote, is inactivated selectively. And in placental mammals, that includes us, only one X chromosome is randomly inactivated while the other one is active. As far back as 1949, the presence of this inactive X chromosome was visible inside cells near the nuclear wall. And that was first discovered by somebody called Murphy Barr. Therefore, sometimes that shut down X chromosome, which is condensed as heterochromatin, is visible and is known as a Barr body. Incidentally, if a female has 3 X chromosomes, in order to do compensation for dosage, 2 of the X chromosomes will be shut down, and therefore in those individuals there will be 2 Barr bodies. Keep that in mind, please. In 1961, the process of Barr body development was hypothesized by Mary Lyon. So she's given credit for the process, the hypothesis, called random X chromosome inactivation hypothesis. During development, a signal is sent that inactivates in cells a random X chromosome. So in some cells, one X chromosome will be randomly inactivated, in others, the other one. This process is permanent. Once an X chromosome is deactivated, it is then inherited in subsequent cell divisions through mitosis. So female mammals are mosaics of 2 populations of cells, as indicated in figure 326. So some tissue has one X chromosome inactivated, and other tissue has alternative X chromosome inactivation. In most cases, this will not be a problem unless she carries 2 alleles for some important genes. We'll come back to that in a second. Both alleles from both chromosomes will be expressed approximately equally over the entire body. The phenotypic effects of this process are apparent in some creatures. For instance, in cats, we have the X chromosome carrying a gene for coat color. And if a cat happens to inherit 2 alleles on her 2 X chromosomes, one for black and one for orange color, then because of the process of lionization, X chromosome inactivation, she is a heterozygote in which some cells will express the orange and other cells will express the black allele. The result is a tortoiseshell cat, also known as a calico cat. A tortoiseshell cat shows you the pattern of X chromosome inactivation is random. No 2 cats are identical. A word of caution though, not every single gene on the X chromosome on the bar body will be inactivated. Some genes, for some reason, are still active. So we have to be aware of that. This random X inactivation requires an X-linked gene called XIST, X inactivation specific transcript. And that gene produces RNA, and that RNA, which are large molecules, they spread out over the chromosome that's expressing that gene and cover the chromosome, therefore inactivating it, making it inaccessible to proteins. And that's known as chromosome painting. This process is only going to happen on the chromosome that generates this type of RNA. And that in genetics is known as a cis form of action, as opposed to trans, which means the opposite chromosome. This is a cis form of activity. Thank you.